Hi friends, welcome to BAMU'S Biogenius. Today we will discuss about cladistics. This topic is related with taxonomy and animal diversity, and it is very important for UG and PG students. So stay with me for cladistics. Now before going to the history of cladistics, we know that Darwin was the first man who speculated that all the organisms or whatever the diversity that is present on the earth that is related with each other, and that's why he um, wrote on a piece of paper this diagram, and this is known as a tree of life. And he believed that the all organisms have a common origin. After Darwin in 1950s, two notions were there for classifying the organism. One is called as the notion, or which believed, or the people or scientists, they believed that the similar individuals, they can be grouped in a particular kind of category. Or we can say that by looking at the phenotypic characters, by looking at the similarities in between the morphological features of the organisms, we can classify the organisms into different groups. So this concept is known as a phonetic concept, which is based on like begets like. Then the another concept, with the advent of science, new methods are used, and by the time 1950s, the Mendel's laws were there, and people believed that, or people came to know that genes are there present, and each, or whatever the phenotypic characters that are there, that are because of the genes. So another kind of we can say that notion that prevailed at that time, that is known as a genealogical classification. And this man was the first man who systematically tried, or scientifically tried, to use that genealogical concept in his book that is known as Phylogenetic Systematics in 1966, where he first described how a phylogenetic tree can be made by looking at the various types of characters, and he devised a method that is known as a cladistics. So, and this method of cladistics devised by Willy Hennig drastically changed our understanding of the relationship among the organisms that are present on the earth. And that is why some of the biologists, they are of the opinion that this work of organizing biological species on the basis of genealogy can be equated with the formation of a periodic table in a chemistry. That is why some biologists, they believe that Willy Hennig deserves the position at the level of Aristotle, Carl Linnaeus, and Darwin in biology. Now, question comes in our mind that why there is a need of a cladistics, why there is a need of another taxonomic classification system when systematics is there. We know that in a systematics we use evolutionary relationship among the organisms to construct a phylogenetic tree. Then why cladistics is needed? See, when we construct a phylogenetic tree by looking at the evolutionary relationship in systematics, we consider similarities and dissimilarities, or we can say that we consider morphological features to depict the phylogeny. Because we know that all the organisms that evolved from each other, and that's why they are similar. But evolution is not so simple. Evolution may be forward, that may be a rapid, that may be a backward, that may be divergent, that may be convergent. So it becomes difficult to correctly make a phylogenetic tree on the basis of only morphological characters or phenotypic features. And for example, you can see here these two organisms, one is a dolphin and other is a shark. If we look into the morphological features of these organisms, we can or we may put them into one group. But in reality, we know that this is a clear-cut case of a convergent evolution, and dolphin is a mammal and a shark is a fish. So they belongs to a totally different groups. Now, another example of a taxonomic puzzle which was solved by the genealogical study is the taxonomic puzzle of giant panda. Earlier scholars, they used to believe that giant panda is related with the red panda because of the similarities they are having. Some of the scholars, they are of the opinion that giant pandas are similar with the bear. So this type of confusion of a taxonomic status of giant panda was remained for more than 100 years. Some of the scholars, they believed that giant panda is similar with the bear. That is because it has a similar large skull like a bear. Giant pandas have also a brain structure similar to the brain of a bear. Even the lung structure is also similar with the bear. But some are of the opinion that these giant pandas are related with the red pandas because of the structural similarity. Apart from size only, the structural similarity was there. The distribution of these red pandas was in the vicinity of these giant pandas. Their diet was similar. In giant panda exclusively feeds on bamboo and bamboo shoots and bamboo leaves. Similar diet was observed in case of red panda. Then even the giant pandas have a opposable thumb. Similar type of structure is also observed in case of red panda. So that means all these kind of similarities were observed in between the giant panda and red panda. So that's why a kind of confusion was there about the taxonomic status of a giant panda. But genealogical studies, that means DNA studies, proved that giant panda is more or less similar with the bear, and red pandas, they are similar with the raccoons. So this is how that this taxonomic puzzle of a giant panda was solved by the genealogical studies. So that's why genealogical studies are very important in identifying the status of various organisms, and that's why it is used in a cladistics. Now, as we know that in every scientific method, certain kinds of assumptions are necessary. Here, in case of cladistics also, we know that cladistics is the scientific method. It is a kind of a hypothesis that predicts a relationship between the organisms under study. And for that purpose, we have to make certain kinds of assumptions. And in cladistics also, there are three basic assumptions that are required. Now, we will discuss these assumptions one by one. First one is any group of organisms are related by descent from a common ancestor. As we know that life originated on earth only once, and whatever the diversity that we see today is a product of their ancestors. And the second one is there is a bifurcating pattern of cladogenesis. You can see here whatever the organism that is present, when it reproduces, or we can say that when this particular species give rise to two other species, it bifurcates into two. So in a cladogenesis, it is always assumed that there is a we can say that splitting of one species into two. Then third one is change in the characteristics occurs in a lineages over a time. So as time passes, the particular kind of a species is not static, but it moves, or it shows a kind of a variation in a next species, or in a subsequent generations, and so that a kind of change that accumulates and that give rise to new species. So this is the third assumption, that is a change in characteristics occurs in lineages over a time. Now, when this such a thing happened, the original character or the primitive character is considered as a plesiomorphic character. So here, in this case, for example, this is an imaginary species, and black eyes are considered as a plesiomorphic character. Now, this species gave rise to two, or bifurcates into two species. Now these have, you can see here, these new species have different types of color in their eyes. So these are the derived characters, and these derived characters from the primitive character. This is a derived character. So such a type of character is called as a apomorphic character. Here in this case, white, red, yellow, and blue eyes are the apomorphic or derived characters from the parent or a primitive species. So this is how that these assumptions are used in a cladistics. Now we will discuss about what is cladistics. Cladistics is a modern system of classification which uses some characters which are ancestral, or which may be derived in a particular kind of a taxon. So it is a method of classifying organisms and grouping these organisms on the basis of shared characters or a unique characters. Then it is called as a cladistics. Now you can see here it is made up of a tree like a diagram. So you can see here it shows a tree like a branches are there. See, several branches are there. So at the end of each branch, a new species is there, and when these branches they merge together at one point, then this is known as a node. Now every node depicts a speciation event. So such a type of a diagrams which shows a relation between the different species and forms a tree like a structure, it is known as a cladogram. Now we will discuss how to build a cladogram. See here, when we are building a cladogram, we have to consider a particular kind of a group of which we have to make a cladogram. Now we are considering here a group of amniote vertebrates. Now, what do you mean by amniot? Amniot means a kind of a cavity which is present around the fetus in all these kind of groups. That is in case of reptiles, birds, and mammals. Now, then we have, if we want to make a cladogram with these, then consider a character which is variable in the group. Now, we have to take a particular kind of a character which is variable in this group. So we will take here the presence or absence of a teeth. Because why we have taken this? Because mammals are also having this, reptiles are also having this teeth, and birds they don't have the teeth. So you can see here, this is how we can connect the reptiles and mammals. And you can see here, this is a node. Now, what this node specifies? This node specifies that the mammals and reptiles must have, or their ancestor must have the teeth, and that's why we are joining these reptiles and mammals. So then the next step is we have to polarize the character. Now, what do you mean by polarization of a character? Polarization of a character means what? We have to determine whether this particular character which is we have taken, that is presence or absence of a teeth, is a ancestral or a derived character. If it is ancestral, then we will have to put this on before the common ancestor. So we have to put this here. If it is a derived, then we have to put this here or here. So we know that the ancestors of reptiles and mammals have teeth. So we are putting this particular character presence of teeth on this particular before the ancestor or before the common ancestor. So this is how that we have to polarize the character. Then the next step is determine that whether this character is shared by the recent ancestor in this group. Now, most recent ancestor in this group is a mammal, so it shows the presence of a teeth. So it is we have already connected it. Then consider that the reptiles. We know that reptiles also have the teeth, but birds don't have a teeth. Now we know that the reptiles that and birds have a common ancestor, and from that common ancestor birds arose, so they lost this teeth secondarily. So here we are putting this absence of a teeth on this particular line, that is before reptiles. So it suggests that the birds have lost their teeth during the evolution. Then these are now we will discuss about the some important features of a cladogram. Now it has ancestral characters at the bottom of a cladogram. I have told you that, or we have discussed already, that whenever we make a cladogram, it is always at the bottom of the um, cladogram. We will get the ancestral characters, and we will get derived characters at the end of the, or we can say at the top of the cladogram. So hairs, these are present only in mammals. So this is a derived character. So that's why the hair, it is present here on the top of the cladogram. Then shared derived characters are present in most recent common ancestors. So you can see here the shared derived character, whatever it may be, that is present on the most recent common ancestor that we all know. Then even if, or we can say that cladogram can be, or cladogram is useful not only for such kind of a morphological characters, but it can be useful when we consider, or in a cladogram we can consider a character related with the behavior, or you can use molecular data to construct a cladogram. So these are all the features of cladogram. Then another peculiar feature of a cladogram is that each small clade is nested in a larger clade. So you can see here, this is one clade of reptiles, birds and mammals. But we can make another clade also, larger clade also, that is that includes amphibians also. So here the all these four have a primitive character of four legs. So from this onwards we can have this orange colored larger clade, and in that orange color larger clade, a smaller clade, or we can say that pink colored smaller clade is nested. So this is how that it is always there that small clade includes, or it is covered by, or we can say that a larger clade includes the smaller clades. Then another peculiar feature of cladogram is that there is a comparison in between, or there should be a comparison between the outgroup and in-group. In-group means the clade, or we can say the group under the study, it's called as a in-group, that we will see in the next slide. Now we will talk about outgroup in a cladistics. Now what is outgroup? Outgroup means a kind of a group or a species which is diverged before the study group. Study group here is called as an in-group. So if we are studying a particular kind of a cladogram, and in that cladogram, if we are studying this particular group, the group or the species which is derived or diverged before this group is called as a outgroup. So this is how that there is a kind of a relationship in between the in-group and out-group. Now when we are studying this in-group, what happens that one kind of species which is not within this group is required to compare, and for comparison this outgroup or a species is considered which is diverged before this in-group, and that's why such type of the species or outgroup is used for comparison. Now how this particular outgroup is chosen? This is chosen on the basis of morphology, paleontology and embryonic development or gene sequences, so that we can compare this particular outgroup with the in-group members. Then outgroup characters are very carefully considered, or we can say that very carefully chosen, so that they should be compared with the members, with the characters of the members of in-group. So this is the importance of the outgroups in cladistics. Now we will discuss about types of groups in cladistics. First one is a monophyletic group. Monophyletic group means a group which contains most recent common ancestor with all of its descendants. Then it is called as a monophyletic group. You can see here most recent common ancestors B and C, and its common ancestor A. So all these are in a packed in a one group. Then such a type of a group is called as a monophyletic group. Then paraphyletic group, when a group contains ancestors but not all of its descendants, then it is called as a paraphyletic group. You can see here a C which is a descendant of this ancestor A, which is not included in this group. So this group is called as a paraphyletic group. Then last one is a polyphyletic group. Polyphyletic group means what? When a group do not contain common ancestors. So all the common ancestors are excluded, and only the descendants are there, and these descendants are unrelated with each other. They don't have a common ancestor. So such a type of a group is called as a polyphyletic group. Now we will discuss about the terminologies in cladistics. First is a plesiomorphy. Plesiomorphic character means a character which is present in the ancestors, or a character which is a primitive type of a character. Then it is called as a plesiomorphic character. You can see here, this A is here is a ancestral character, because all these taxons, they show this particular ancestral character A. So you can see here, in each and every taxon you will get the plesiomorphic character. Now apomorphic. Apo means derived. So derived character means apomorphic character. So here in this original taxon, and this AC taxon is derived from this original taxon. Now this C is a apomorphic character because it is newly formed. So that's why derived character, it is called as the apomorphic character. Now when this odd apomorphy. Now next is odd apomorphy. Odd apomorphy means the character which is present only in one taxon, then it is called as a odd apomorphic character. Then syn apomorphy. Syn means shared. Apo means derived. So you can see here, a shared derived character is called as a syn apomorphy. You can see here, C is shared by these two taxons. So that's why such a type of character is called as a syn apomorphy. Now, for example, forearms bones of humans and cat, they are syn apomorphic characters. Then symplesiomorphy means, you can see here, sim means shared, plesio means ancestral. So when ancestral character is shared by the taxon, then it is called as a symplesiomorphy. Example is quadrupedalism in mammals. So you can see here, symplesiomorphy, ancestral character, this is a plesiomorphic character, but this is shared by all the taxons. So that's why A is called as a symplesiomorphic character. And example here is a quadrupedalism in mammals, because we know that from amphibians onwards you will get the quadrupedalism. So that's why this quadrupedalism character is a shared ancestral and shared character. Then homoplasy. Homoplasy means the character which is shared by two different taxa, but it is not present in their ancestors. Then such characters are called as a homoplastic characters. The phenomenon is called as a homoplasy. Example here is wings of bird and bat. So these are the terminologies in cladistics, which are very simple. Just you have to remember plesio, that means ancestral, apo means derived, syn or sim means you have to remember that it is shared. So nothing else. Now we will discuss about the applications of cladistics. First application is, it is useful in creating system of classification which is based on evolutionary theory, genealogical theory. Second application is, it has a power of prediction. When we use cladistics, it can be used scientifically to predict the relationship among the taxa. Third one is, it helps in explaining mechanism of evolution, how the character, a particular kind of a character evolved in a particular taxon, that can be traced with the help of cladistics. Then not only in case of biology, but in case of other studies, other non-biological fields, for example, linguistic studies, cladistics can be applicable. So we will see in detail about these applications. Now the first application of a cladistics is it is a very useful tool in creating system of a classification. Paleontologists found it a very helpful in classifying the vast diversity of organismic world because it uses a evolutionary theory, and every significant evolutionary step is marked as a bifurcating branch that leads to a sister taxa which is genealogically different from each other but at the same rank. It also, or this type of a classification also uses a recent common ancestor to relate with the other taxa. Now, the second application of cladistics is the power of prediction. It has been found that cladistics is very helpful in fixing the position of animals or showing the relationship of among the animals with the help of certain characters. These characters may be morphological characters or the anatomical characters or genomic characters, which is based on DNA sequence, RNA sequence, or the sequence of amino acids in a protein. We know that the position of whales or the relationship of whales is a long-standing mystery. It was debated in a scientific circle for at least 40 years. Although Darwin predicted the terrestrial origin of whales, but fossil evidences were not there. Even some characters of its terrestrial origin were present in the whales, like presence of lungs, then presence of whiskers in on a young born, or the development of hindling in a fetus. All these were suggesting the terrestrial origin of whales, but still, because of the lack of fossil evidence, this particular origin of whales remained mystery. Then it has been also found that all the mammals or all the ungulates that are present today, they are having a particular kind of a bone in their hind limbs that is known as ankle bone. These ankle bones are of two types. In some ungulates, it is a single pulley bone astragalus. In some ungulates, it is a double pulley bone astragalus. In case of even-toed mammals, or we can say the Archaeodactyla group, the ankle bone is a double pulley bone. Now, such a type of ankle bone is not observed, or it is very useful in the forward mobility or a greater mobility on the land, and such type of ankle bone is not observed in the whales. So on the basis of presence and absence of this ankle bone, a hypothesis is developed and is predicted by this cladogram, which suggests that the this absence of this pulley shaped bone may be a primitive character and present in the ancestor of all these land vertebrates. And it is also predicted that later on, this particular land vertebrates or ancestor of land vertebrates gained this pulley shaped bone for greater mobility on the land. But with the advent of molecular biology tools, some molecular biologists sequenced the amino acid from the hemoglobin of land vertebrates and compared it with the whales. And what they found that the whales hemoglobin is similar with the hippopotamus hemoglobin. And then they revised this cladogram like this and placed whales alongside the hippopotamus in the group of even-toed animals. But still, paleontologists were not convinced. So this mystery remained as it is. But later on, in 2001, the whale fossil bones were discovered in India and Pakistan. These are the regions where these particular whale bones were discovered, fossil whale fossil bones were discovered, and it has been found that in some cases like this is a fossil of early relative of a whale, Pachycetus. It has been found that the ankle bone astragalus is present in these animals. That means the whale's ancestors have this ankle bone, which suggests the common ancestry of all the land vertebrates. And again, it revised the cladogram into such type, or we can say that although all these animals, Artiodactyla group or even toad animals, have this pulley-shaped bone, it was lost secondarily in case of whales. So, and this is how that the cladogram is again revised and it confirmed the position of whale alongside the hippopotamus. So this is how the cladistics is very helpful in predicting the position of organisms in a taxonomy. Now the third application of a cladistics is it is very useful in explaining mechanism of evolution. For example, in case of spiders, there are two types of spiders, orb weaving or orb web spiders and cobweb spiders. People used to believe that cobweb spiders or a cobweb building behavior is a primitive kind of a behavior, and looking at the complexity of orb webs, these are all called as orb webs. So looking at the complexity of the orb webs, it has been speculated that cobweb behavior is more primitive and orb web weaving behavior is a more recent one, and that's why people used to believe like this. But now, with the help of cladistics, it is clear that the cobweb building behavior is considered as a more recent one as compared to the orb web weaving behavior. So this is how, for explaining mechanism of evolution, cladistics is also important. Now we will discuss the demerits of cladistics. The first demerit is two or more lineages can arise from a single population. Such type of phenomenon is called as a polytomy. You can see here, as in case of cladistics, what is assumed that at each node or for every evolutionary state, a bifurcation is there in a lineage, but it may be possible that from that single um, lineage, two or more lineage can also be produced, can also be evolved. So such type of event is called as a polytomic event, and it is also possible. But cladistics supports only bifurcation of lineage. So this is the demerit of cladistics. Then cladistic methods cannot show the interbreeding between the two distinct groups. So whenever we talk about the cladogram, cladogram does not involve the interbreeding, or we can say it cannot show the interbreeding in between the two distinct groups. So that is also a kind of a demerit for cladistics. Then the last one is the cladistic method cannot be applied to organisms developed from lateral gene transfer. As we know that new organisms can also be formed in case of bacteria also. You can see that lateral gene transfer that takes place, and if that particular lateral gene transfer takes place, then cladistics methods cannot show such a type of exchange of genetic material in between the two distinct lineages, and that's why the cladistic method is also failed here to demonstrate the lateral gene transfer or horizontal gene transfer. Another demerit of a cladistics is the lack of sufficient available distinguishing characters. As we know that whenever we draw a cladogram, we look for distinguishing characters in between the taxon, and on the basis of that particular polarizing characters, we shape our cladogram. So it most of the time what happens that because of the insufficient distinguishing characters, we could not properly classify or distinguish the taxon. So this is one of the reason that is a demerit for cladistics. Then next one is there is the uncertainty as to which characters are ancestral and which are derived. So most of the time we could not identify which type of characters should be considered as ancestral and which type of characters should be considered as the derived characters, mostly in case of fossils. So that is again another kind of demerit in devising a proper cladogram. Now the last demerit is the difficulty of a clear recognition of convergences. As we know that convergent evolution means the appearance of a particular type of or a similar type of a characters in two very different types of lineages which are not related with each other, but still they are having the similar type of characteristic features. So such type of characters, or so in such type of a taxon, we cannot make a cladogram with this type of convergent entities, or we can say that two lineages which shows a convergent evolution. So these are all the difficulties in cladistics. Now, according to the philosopher Karl Popper, for a theory to be scientific, the whatever the hypothesis that is presented must have certain types of characteristic features or criteria. First one is that hypothesis should be falsifiable. So whenever a cladistic method is used, it is nothing but a kind of a, whenever a cladogram is used, it is nothing but a kind of a hypothesis, and which is based upon the availability, or we can say that by observing a particular kind of a traits in that particular group, and on that basis the cladogram is prepared, and it is also falsifiable. Then cladogram has a predictive value, so it can also predict the relationship among the various taxons. And the third one is it has a explanatory value. Whenever we draw a cladogram of a particular type of a group, it has a explanatory value. It explains how all these organisms in that particular clade are related with each other. So on that basis, we can say that cladistic method is a kind of a scientific method, and that's why it is popular even now also. So this is about the cladistics. Thank you very much for patient hearing. Please subscribe to my YouTube channel BAMU'S Biogenius for latest uploads, or like my videos and comment. Thank you.